Namaste students. हमेशा खुश रहो बिटिया. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth Women's University, Jaipur. So today we are here to discuss the topic that is downstream processing. Okay, so this topic comes under the subject upstream and downstream process, and we have already learned about the upstream processing. Okay, so next move towards the downstream processing. So what is the definition of downstream processing and what is it? So downstream processing refers to the recovery and purification of biosynthetic products from the natural sources such as the animal or plant or from the fermentation broth, and the proper treatment and disposal of waste is also included in the downstream processing. So basically downstream processing is the collection or the isolation of the product. So this product can be isolated from various sources. Okay, the source may be the animals or the plant or the culture fermentation, and in the downstream processing, along with the collection isolation of the product, the disposal of the waste, proper disposal of waste is also included. Okay, so there it is an essential step in the manufactures of the pharmaceuticals. Okay, such for example, when the antibodies are produced, where the hormones are produced like the insulin and other growth hormones, uh, and the vaccines are produced, the antibodies are produced. uh, so in the industries or in the practicals, this is an essential step. Okay, because it is the last step where you can recover your product, and the quality and quantity of your product depends on the downstream processing. Okay, simultaneously you have to take care about the disposal of the waste you have generated during the upstream and downstream processing. Okay, so there are few stages in the downstream processing, step by step processes that how you can uh, collect your final product. Okay, so basically the four stages are there. um, Cell ferment, tissue cell and fermentation broth. It is used for the progressive purification and concentration of the your culture media or the plant material or the animal tissue. Okay, so first of all, the step is the removal of the major insolubles. Okay, so in the first step, you capture your product and the as a solute in a particle free liquid. Okay, for example, the separation of cells, the separation of the cell debris and other material which is found particulate as a particles in the fermentation broth or in the fermentation media. Okay, because if suppose there is a hormone or antibiotics you have produced, and the using the bacterial cells, so you first you have to isolate the particulate bacteria or the cells. So the typical operations to achieve this are the filtration. Okay, you can filter out the insolubles. You can use the centrifugation mechanism. You can use other chemical sedimentation or chemical precipitation methods, and the other precipitation like the using the this natural gravity. You can wait for some time, the insolubles will the precipitate down, and you can recover the first raw material from the uh, upper part of the culture media. Then the second stage is the product isolation. So it is the removal of those components whose properties vary considerably from the desired product. Okay, for most of the products, water is the chief impurity. So the isolation steps are designed to remove the most of the water, so reducing the volume of material to be handled and concentrating the product. So the solvent extraction, absorption, the ultrafiltration, precipitation are the some techniques from which you can concentrate your material and remove the extra water. So next is the product purification, that is done to separate those contaminants that mimics like the product. Okay, so that are chemically and physically very close to the properties of the your product. So consequently, steps in this stage are expensive to carry out and require sensitive and sophisticated equipments. So this stage contributes a significant extraction of the entire downstream processing expenditure. So example is they include the affinity and size exclusion chromatography, reverse phase chromatography, and the other chromatography techniques, the crystallization and the functional precipitation. So final step is the product polishing. So it describes the final processing steps, and that ends with packaging of the product in a form that is stable, easily transportable and convenient. So the desiccation, the lyophilization, the spray drying, and the crystallization techniques you can use here. So this depends on the product and its intent and use. So polishing may also include operations to sterilize the product and remove the deactivate trace contaminants. Okay, so for the safety purpose of the product. So the such operations might include the removal of the viruses and the bacteria, which may create problems in the product. Okay, so these are the main steps, but there are the setups in the industries which can merge or increase the steps during the product purification. Okay, so thank you. This is I hope you are satisfied with this detailed session, and if you have any query, please write in the comment box. uh, and this is powered by digital version 2 Jyoti Vidhyapeeth Women's University, Jaipur. Thank you.